Hi friends, how are you? I hope you are enjoying my videos and my site logicalbiology.com where I am writing the interesting articles on biology. So in this video I am going to explain the annexine 5 and propydium iodide staining of the cells, which is a very important technique in the field of cell biology where we want to detect whether the given cells are doing apoptosis or not. So to detect apoptosis, we are using which staining? Annexine 5 and PI, and this technique we are using for the detection of apoptosis in the given cells. Now, whenever we hear the word apoptosis, then we get the definition that is a programmed cell death, which is a cell death pathway which is used to remove the unwanted cells. And this is very important in development of organs, development of nervous system and development of immune tolerance, or whenever you want to remove the cells naturally, right? But if you are a researcher where you are studying, development of cells or differentiation of cells or effect of drugs on different cells, or you are working on a drug which you are suspecting that can kill the cell, it is an anti-cancer drug. In that case, you would like to detect the apoptosis in the given cell population. Now, if you want to detect a natural pathway, then you have to have little bit knowledge about that pathway, so you can take the advantage of that situation, right? So whenever I am talking about a healthy cell, then healthy cells are having a phospholipid that is called as phosphatidylserine, and this phosphatidylserine is present inside the cell membrane. And if this cell is entering in the apoptosis pathway, then this phosphatidylserine is flipped out of the cell, and finally when the apoptosis is done, then it becomes dead cell. So I can divide the apoptosis in the whole pathway in three parts. You can have healthy cells where the phosphatidylserine is present inside. You can have the cells which are live but already entered in the apoptosis. In that case, the phosphatidylserine is flipped out of the cell, and finally you are having the dead cell. So we are going to take the advantage of this situation in a way so we can detect the apoptosis in given cells. So how we are going to achieve it? So we are having a very important protein which is called as annexine 5. Now the speciality of annexine 5 is it is having very high affinity towards phosphatidylserine. It means it can bind to phosphatidylserine in an efficient manner, and we can create a fluorescent conjugated annexine 5. So one of the chemical or one of the molecule we are using in this technique is fluorescence conjugated annexine 5, which is having its target as phosphatidylserine. Second, we can use a dye, a propydium iodide, and propydium iodide is a DNA binding dye, but the special part is it can bind to the DNA in dead cells, and again it can give fluorescence. So in this technique we are using two fluorescent molecules. One is fluorescent conjugated annexine 5 and second is propydium iodide. Right, now I am again going to dissect the whole apoptotic pathway when we will mix the cells with fluorescent conjugated annexine 5 and PI. Okay, so we are having viable or healthy cells where the phosphatidylserine is present inside. So when I mix the annexine 5 and PI, from the viable cells I will not get any fluorescence. Why? Because phosphatidylserine is present inside the cell, so annexine 5 cannot bind, as well as cells are live, so propydium iodide cannot go inside and can bind, right? Once these cells are entering into apoptosis, then what is happening? The phosphatidylserine is flipped out, and once they are flipped out, then mixed annexine 5 can go and bind to it and can give us the fluorescence. And finally when the cells are dead, so the phosphatidylserine which was flipped out is already bound with annexine 5, and also propydium iodide has entered inside the cell, bound to the DNA and have given the fluorescence, right? So viable cells, no fluorescence. Cells which are live now but currently doing apoptosis, they will give fluorescence only for annexine 5, and cells which are dead because of apoptosis, they will give fluorescence for both, that is fluorescence for annexine 5 and fluorescence for propydium iodide. So three types of populations you can have: viable cells, double negative fluorescence, and currently doing apoptotic means cells which are doing apoptosis currently, but till now they are not dead. We'll be getting single fluorescence for annexin 5, and dead cells will be getting double fluorescence for both annexin 5 and propydium iodide. So when we analyze the cells by flow cytometer, then we can make a graph, and on the x-axis of graph we can keep the fluorescence intensity for annexin 5, and on y-axis of the graph we can keep the fluorescence intensity for propydium iodide. Now we will be having 4 quadrant, 1, 2, 3, 4. So in this quadrant the cells are annexin 5 negative and pi negative, so we'll be calling them as healthy cells. Then in the next quadrant the cells are annexin 5 positive but still pi negative. Then these cells are doing apoptosis but still live. Now in this quadrant the cells are double positive, they are positive for annexin 5 as well as they are positive for pi. So these are dead cells and the reason of death was annexin. So how we are deciding it is positive negative? So it is simple quadrant graph where you have double negative quadrant, then you are having single positive quadrant, another single positive quadrant, and then double positive quadrant. And this kind of graph you have studied at so many positions, and similar kind of graph you also have the Ramachandran plot where you are having 4 quadrants and you are giving different values. So this kind of graph we get when we are running the cells in flow cytometry after mixing annexin 5 and pi. So I show you a live means the real life experimental graph. So here we are having the graph. So on x-axis we are having annexin 5 fluorescence, on y-axis we are having propydium iodide fluorescence. So these are the cells which are double negative, so these are live cells, and these are the cells which are only annexin 5 positive, pi negative, these are early apoptotic cells or the cells which are doing apoptosis, and finally the dead cells which are positive for both annexin 5 and pi. And in flow cytometry, for your information, one dot is representing one cell, and whenever you are having this word FITC, so FITC is a fluorescent conjugate which is bound with annexin. So FITC is generally giving green fluorescence and propydium iodide is giving red fluorescence. So it is very simple, and the software can calculate the percentage of cells. You can tell how many cells are viable, how many cells are early apoptosis, and how many cells are in late apoptosis. So it was very easy technique, but in so many entrance exams and in so many interviews people asked about this technique. And also this technique is very important for research purpose. For example, if I am treating the cells, the cancer cell lines, with a anti-cancer compound or anti-cancer drug, then I will be having 2 sets of cells, control cells where drug is not added and test cells where the drug is added. So I assume if I am lucky, then I am going to have more cells in these quadrants, which means the drug is inducing the apoptosis, and in control most of the cells will be in this quadrant, it means they are healthy. Getting my point? So there are many applications where you have to find the percentage of apoptosis doing cells, and that can be done.
Hello, I decided to post a small lecture for you concerning flow cytometry in the hopes that this will help you with the upcoming lab that is happening this week. So, flow cytometry is a technique that I have used throughout my graduate career and postdoc, and it's a very powerful technique that allows us to do a couple of things. Number one, it will allow you to quantitate cell populations based on their size, their internal complexity, and also the expression of cellular markers. We can look at proteins, we can look at lipids, we can look at DNA, RNA, you name it. As long as you have a fluorescent marker that you can use to label that particular marker, you can see it by flow cytometry. In a sense, it is very similar to fluorescent microscopy. If you have a reagent that can fluoresce and be used to identify the molecule, you can use it in flow cytometry. Now, because of the complexity of the instrument, it also allows us to separate cells. So we can, some instruments called sorters are able to allow us to separate cells based on the expression of a specific cellular trait. For example, you can look at a cell that it's positive for the expression of a protein and isolate that cell and separate it from a population of cells that is negative for the same expression of a protein. Or you can look at cells in the cell cycle like we're going to do in the lab, and let's say isolate the cells that are in G1 or G2. So you can separate them to about 95% purity. So it's a really reliable technique to allow us to do a lot of different samples. In this case, what we're going to be doing is to examine cell cycle stages, and we're going to measure the amount of DNA present in each cell. We're going to have a DNA staining agent that is fluorescent, and therefore we're going to be able to measure the amount of DNA present in that cell. So in a sense, since we're measuring fluorescence and you have a direct correlation with the amount of fluorescence to the amount of the reagent, in this case DNA, the greater the amount of fluorescence, therefore the greater the amount of DNA that the cell has. So now, the instrument, it's very complex, and my professor at Stanford who invented this instrument really had a team of engineers working with him that helped them bring in together a lot of different things. So one of them is cell sorting and flow issues, and the other one is mixing that up with cytometry. So what we have usually, it's a chamber called a nozzle, and in the nozzle you have two different fluids that are going to be sort of mixed together. So in the nozzle you have a sheath fluid that is going to be surrounded the cell suspension containing the cell sample. That is going to force the cells, which are usually chaotically mixed in the solution, to go in a single file. That is hydrodynamic focusing. So you're going to, because of the shape of the nozzle, focus the cells that are usually, like I said, all mixed up into a single file structure. At that point, what you have is the questioning or the identification of the cells. Since we're looking at a fluorescent marker, we need to stimulate that marker with light, and in this case we're using lasers. So as the cells are passing single file through the stream, they're interrogated by the laser. And as any particles that are hit by light, they're going to be able to scatter light. Some scattered light will be scattered in the same direction of the laser, we call it forward scatter, and other light is going to be scattered 90 degrees from the direction of the laser, and we call that side scatter. All the fluorescence also is going to be measured at 90 degrees from the laser. And what we have now in the instrument, just like we did in the fluorescent microscope or the confocal microscopes, are detectors that are going to be able to visualize and detect the light. That light is going to be converted into electrical impulses that the analyzer, the computer at the end, is going to be able to allow us to see as one event. Now, in the case of the sorters, the nozzle, it's vibrating, as I said over here, by ultrasonic vibration, meaning that it's vibrating very, very fast. And what that is doing, basically, it's to get the stream that is coming out of the nozzle and breaking the stream into small droplets. Each droplet contains one cell. So when the droplet begins to separate from the stream, you can give that droplet a particular charge. And therefore, you can now have droplets that are negatively charged, other ones which are positively charged, and some of them that are neutral. At the end, you can pass those drops by a very powerful magnetic field, and you can now separate the cells into different components depending on the charge the droplet has. So for example, in this particular example, you have the cells that were red, and those are being the droplets were positively charged and therefore deflected to the negative pole of the magnetic field. The black cells were in droplets that are negatively charged, and those are being directed towards the positive side of the field, and the droplets that are neutral go into the middle on a wastebasket. So in that regard, the instrument that we have at class, it's going to allow us to visualize the cell and measure the fluorescence analysis of it. So you're going to be able to just take the sample and see what the cell's population is going to be in respect to the DNA. We're going to mark the sample with a dye called propydium iodide, and that dye binds to DNA. It actually intercalates DNA, which means that it goes in between the bases, and the more amount of dye that is present, that means that more DNA that is present. So just as an example, just remember that we're going to be looking at cells in different stages of the cell cycle. And because those cells are at different stages of the cell cycle, they're going to have different amount of DNA. So at the beginning of the cell cycle, cells are in the G1 state. G1 state are cells which have basically just divided. and they're initiating the growth phase to become larger. Once they pass the G1 stage, the cells enter the S phase, and the S phase is where the DNA is going to be replicating. So therefore, we're going to have now the chromosomes beginning to replicate. So the cell which are at G1, which are usually have a ploidy of N. So think about the number of chromosomes present in G1. So that's, let's say, in a cell that has only 10 chromosomes, and it's going to be, the number of 10 is going to be equal to 1, because you only have 10 chromosomes. So that will be the ploidy of 1n. That cell, when it's beginning to divide, you're starting to now make copies of the DNA, increasing the size of the DNA inside the cell. Once the DNA has been completely replicated, that cell has double the amount of DNA inside of it. So each one of those 10 chromosomes now has a twin sister, and therefore the ploidy is 2n. Later, during the M phase of cell cycle, the cell will be able to segregate the chromosomes, and each daughter cell will have then an equal number of chromosomes. So therefore, at the end of the M phase, the cells go back to G1 and therefore go back to a ploidy of N. When we're looking in flow cytometry, remember that we're counting number of cells. So in the y-axis of this figure on the right, we have the number of cells which are available and being read. And on the x-axis, we have the relative amount of DNA measured by fluorescence. So cells that are in the G1 state, they have a ploidy of 1, therefore all of them have the same amount of DNA, and therefore you have a nice long peak of cells with a relative amount of DNA of 1. Once the cells start to go to the S phase, they are all dividing the DNA independently. So you begin to see an increase in the amount of DNA that is shown here in the middle portion of this histogram. At the end, when the cell has completely duplicated the DNA, now they have a relative amount of 2. So ploidy is going to be 2n. Therefore, the cells which are in G2 and the N phases right before cytokinesis, they have twice the amount of DNA than the cells that are present on G1. The cells which are dividing DNA here in the S phase are beginning to have various amounts of DNA, and therefore you have a very long distribution of cells in the middle between the G1 and the G2 peaks. So this is a histogram. This is showing the number of cells that are falling within having one quantity of DNA here marked by 1 versus the cells here falling that are already that have already gone through G2 S phase, excuse me, and are now present in G2. So they have now 2 N DNA, and then the ones in the middle that have the S phase. So let's take a little bit closer look at some actual data. So this example here from a previous lab shows the cell number in the left and the amount of fluorescence measured by propydium iodine emission. As I mentioned, propydium iodine binds to DNA and therefore fluoresce. So here, marked in 200, this is the relative fluorescent intensity. You have the cells which are in G1, therefore they have only one pair of chromosomes for each of their chromosomes, and they have